A very good afternoon, or good morning, or good evening to everyone. So, most probably, this is our last class on external things external to cell, bacterial cell, and after that, we will move on to the cell wall and structure external to cell wall. So, flagellar ultrastructure. First point is determined by using transmission electron microscope. So when we talk about flagellar ultrastructure, it is only visible under transmission electron microscope. And flagellar ultrastructure is divisible into three parts: filament, hook, and basal body. When we talk about filament, it is the longest part of the flagella, it is the outermost part of the flagella, and is composed of single type of protein unit which is called flagellin, which is 30 to 60 kilodalton, and hollow and cylindrical structure. It is an hollow and cylindrical structure also. Next is hook. This hook connects the filament and the basal body. It is the more wider region and present outside the cell wall. When we talk about basal body, it is the complex portion of the flagella, and it anchoring the flagella to cell wall and plasma membrane. This anchors the flagella to the cell wall and the plasma membrane. In typical gram-negative bacteria like E. coli, basal body is made up of four rings. In gram-negative bacteria, this basal body is made up of four rings. One is called L ring, other is called P ring, next is called MS ring and C ring. LPMSC. These are the four rings in flagellar ultrastructure, part basal body. Now let's see what are these L ring, P ring, MS ring and C ring. So when we see L ring, this L ring, L simplifies, it is attached to the LPS or outer membrane. P ring, it is attached with peptidoglycan. MS ring, it is attached with cytoplasmic membrane. C ring present on cytoplasmic side. So this C ring is present on the cytoplasmic side. MS attached on the cytoplasmic membrane. P attached with peptidoglycan. L attached with LPS or outer membrane. Surrounding to inner ring, MS and C is the inner portion, inner ring portion. Some proteins are found. What are these proteins? Mott protein and flea protein. Mott protein is of two types, Mott A and Mott B. We'll see in details. Also contains central rod that connect all four rings. So there is also one central rod which connects all these four rings. Let's see. Now, when we talk about gram-positive bacterial basal body, it has two rings, outer ring and inner ring. Outer ring is connected to peptidoglycan, like this portion, it was connected to peptidoglycan, and inner ring, it is this inner ring MS and C. This is connected to plasma membrane, like that. You can remember. Now, when we talk about the flagellar ultrastructure of gram-negative bacteria, you see this. Draw this. This is the outer membrane, this is the peptidoglycan portion, and this is the plasma membrane. Now you have L ring, P ring, MS ring attached to the cytoplasmic membrane, and C ring on the site of the cytoplasmic membrane. This is Mott protein, this is Mott B, this is Mott A. This is filament, this is hook, and these are the flea proteins between MS ring and C ring. So this is your flagellar structure for gram-negative bacteria. Okay, next let's see. This is gram-positive bacteria, a peptidoglycan and plasma membrane only present, and there it was outer membrane, peptidoglycan and plasma membrane. This one, outer membrane, peptidoglycan and plasma membrane, and here we have peptidoglycan and plasma membrane. So filament, hook, this is a central rod, outer ring is over here, peptidoglycan and plasma membrane over here, and inner ring is in the plasma membrane. This is gram-positive bacteria, this one. Next, let us see mechanism of flagellar movement. How the flagella moves. Flagella contain motor, And obviously the basal body is the complex portion, so basal body has a motor that generate torque which is transmitted by hook and filament, and by that the flagella rotate. So this motor has two part, one is rotor, one is stator. Rotor contain LP, MSC and central rod, and stator have mod A and mod B protein. The rotation of the flagella is actually explained by proton turbine model. Rotation occur by expense of PMF, proton motive force. So proton movement occur across the cytoplasmic membrane through MOT protein. So this proton movement move through the cytoplasmic membrane like H plus maybe. So this move through the cytoplasmic membrane through MOT protein, and this extend electrostatic force on the MOT protein, on the rotor protein, that driven torque generation. So this exert proton movement through the cytoplasmic membrane through MOT protein. This exert electrostatic force on the rotor protein that driven torque generation. So this is the proton turbine model. There is C ring and MS ring. See, proton turbine model. C ring and MS ring was the internal part. So we have over here C ring and MS ring. Now in this C ring and MS ring, uh, see, you can see distribution of charges. This generate the torque and this will rotate, and this mod A, this mod B which is not needed. So this mod A through the cytoplasmic membrane, the uh, proton flows, and this create electrostatic force which exert the rotor protein to move, and generate torque. So this um, as this charges and as the proton flow rate will increase, the proton motive force, the speed of rotation will increase. Okay, so as this H plus, more H plus will flow, the speed will increase and rotation speed will also increase. Okay, so motor proton motive force will increase. Now when we talk about bacterial movement, first is swimming, occur in liquid medium done by help of flagella. Two type of swimming are there. One is run, other is tumble. Run is counterclockwise movement occur, and smooth swimming, and cell can move from one spot to another. So cell will move from one spot to another. Tumble is clockwise rotation, reorientation of bacterial cell. The bacterial cell, I am explaining it. Like suppose I have done like this. Okay, so this portion is your tumble. The thing is turning. So this turning point is tumble, and when it is occurring, this portion is run. See, I have explained it here. This portion is run. This is tumble, run, tumble, run, tumble, run, tumble, run, tumble, run, tumble. So this way move. Depending on attractant is present, during no attractant is present, when no attractant is present, it goes and again turns into the same position. Okay, run is counterclockwise movement occur, and this is clockwise movement, reorientation of bacterial cell, smooth swimming, and cell can move from one spot to another. But when concentration is present, so maybe chemotaxis towards the chemical, so this will go and move straight towards the chemical. Okay, or photostaxis, if towards the light, then it will go and move towards the light. No attractant, then this way. So this swimming is done by monotrichous and peritrichous flagella. Now when we talk about swarming, it is done by peritrichous flagella. Okay, uh, group uh, move on the plate and show characteristic morphology. Uh, it occurs on the moist surface. So see, you can see the branching pattern, it looks like dendrites. So branching pattern on the agar plate called dendrites. So this kind of movement is called swarming, when you can see a pattern. Now is spirochete motility occur in spirochete family. Okay, spirochete family, they have flagella, but it is endoflagella. The flagella is produced inside of the cell wall and do not extend outside the cell wall. The flagella remains in the periplasmic space and is covered by the outer membrane. Okay, and twisting like external flagella and that cause rigid spirochete to rotate like corkscrew. Okay, twisting like external flagella that cause rigid spirochete to rotate like corkscrew. So you can see over here like this is the flagella. So this way it will twist, like this way it will twist. So inner all this will twist. So this way little it will move. This is called endoflagella. Now twitching motility. Till now all was of flagellar movement. Okay, like this was endoflagella, but it was flagella. But now this is not flagellar. Twitching motility and gliding motility. What is twitching motility? Occurred in solid surface done by type 4 pili by attending attract and extend and retract policy. This movement is jerky, shaky, and short movement, intermittent. Also called social motility in Myxococcus xanthans because a group of mass of cells move together in uh, coordination fashion. Okay, M. coccus also show gliding motility, uh, adventurous A motility, individual motility occur. Flagella like MOT and MOTB protein perform gliding motility. Okay, slime layer also involved in that motility. Example Flavobacteria. So this was your today's class. You can just take a screenshot. There is a swimming part. So it's done.